Welcome to This Explainer. Today, we're doing a deep dive into the biochemistry of the human immune response. We're mapping out the absolutely incredible, highly orchestrated chemical events that kick off the literal second your body realizes, hey, we're under attack. Think about the last time you scraped your knee, or maybe caught a bug, or burned your hand. You know the drill, right? Swelling, heat, maybe a fever, and yeah, definitely pain. But what actually causes all that? To really get this complex biochemistry, we have to ground it in what we actually feel. Those symptoms aren't just random side effects. They're the direct result of microscopic chemical messengers meticulously commanding your body's defense forces. So, here's our roadmap for today. First, we'll meet the inflammatory mediators. Then we'll look at the preformed first responders, followed by the newly synthesized cell mediators. After that, we'll break down the arachidonic acid pathway, and finally wrap up with the plasma-derived complement system. All right, part one. Let's meet the inflammatory mediators. Think of your immune system like a highly coordinated military operation. At the top level, we've got our cell-derived mediators. You can think of these guys as your local ground troops. They're produced right there at the site of injury. And they're either preformed, meaning they are locked, loaded, and ready to deploy instantly, or they're newly synthesized, meaning they have to be manufactured on site after the damage actually happens. Now, looking at the other side, we have the plasma-derived mediators. These are your systemic reserves. We're talking mostly about the complement system and the coagulation cascade, which just float around in your bloodstream waiting for the signal to activate. Moving on to part two, preformed, the first responders. So these are the quick response forces of our immune battlefield. Because they're already stored up inside the cells, they get released the exact millisecond tissue damage occurs. The two major players we need to talk about here are histamine and serotonin. Histamine comes from the amino acid histamine, and it's pumped out in massive quantities by your mast cells. Then you have serotonin. Now, you probably know serotonin as a mood chemical in the brain, but it doubles as an inflammatory mediator here. It's derived from tryptophan and mostly released by enterochromaffin cells. So what do these preloaded weapons actually do? Well, they immediately trigger vasodilation, widening your blood vessels to rush blood to the site. And they cause bronchoconstriction. That narrows your airways to physically block toxic gases or pathogens from getting deep into your lungs. All right, part three, newly synthesized cell mediators. Now we bring in the heavy artillery that gets manufactured on site, cytokines, specifically a group called interleukins, or ILs. And every single one of these has a highly specific job. IL-1 acts as a pyrogen. Basically, it tells your brain to crank up the thermostat and cause a fever. IL-5 activates your eosinophils, while IL-6 regulates acute phase reactants in your liver, like C-reactive protein. IL-8, that one recruits neutrophils straight to the infection site. But my absolute favorite has to be IL-10. While all the other interleukins are screaming for more inflammation, IL-10 acts as the anti-inflammatory hero. It's like the rare commander ordering a tactical retreat, telling the immune system to just stand down because the battle is won and it's time to start healing. Next, let's talk about chemokines. Think of chemokines as highly targeted distress signals. They act as chemoattractants, meaning they recruit very specific, specialized cells to the front lines, all based on their chemical structure. C-type chemokines, like lymphotactin, specifically call in the lymphocytes. CC-type chemokines, like eotaxin, bring in the eosinophils. And CXC-type chemokines, which actually includes our good friend IL-8, are specifically designed to summon neutrophils. There are a few other really vital, newly synthesized mediators we absolutely have to mention. First is nitric oxide. This is produced to cause vasodilation and to stop your platelets from clumping together when they shouldn't. Then there's tumor necrosis factor alpha, or TNF-alpha. Now, this causes fever, but it also drives extreme weight loss, which is known as cachexia, something we often see in severe chronic illnesses. And finally, interferons, alpha, beta, and gamma. What's truly amazing here is how we've actually harnessed this specific biology for modern medicine. Interferon beta, for example, is naturally produced by fibroblasts, but we synthesize it and use it clinically as a highly effective treatment for multiple sclerosis. It's a real game changer. Let's shift gears to part four, the arachidonic acid pathway. This right here is one of the most foundational biochemical cascades in all of human pathology, and it starts right at the site of the damage. So every cell in your body is wrapped in a phospholipid membrane, right? When a cell gets damaged, an enzyme called phospholipase A2 wakes up. It literally chops up those membrane phospholipids and converts them into a massive pool of a chemical called arachidonic acid. You can basically think of arachidonic acid as the raw manufacturing material for the entire next wave of the immune response. From here, this raw material splits off to build two completely different arsenals. It can go down the cyclooxygenase or COX pathway. This path produces prostaglandins, which cause that awful throbbing pain and fever we're all familiar with, as well as thromboxanes, which triggers blood clotting. Or, alternatively, it can go down the lipooxygenase or LOX pathway. This path produces leukotrienes, and leukotrienes happen to be the most potent bronchoconstrictors in the human body, dramatically clamping down your airways. So why does this matter? Well, the crucial point here is how this translates directly to your own medicine cabinet. If you take a steroid, you're actually shutting down this entire pathway at the very top. Steroids inhibit phospholipase A2, completely halting all production of arachidonic acid. But what if you take an NSAID, like ibuprofen? NSAIDs are a bit more targeted. They specifically jam up the COX enzyme. By physically blocking just the COX pathway, they cut off the production of prostaglandins. And that is exactly why popping an ibuprofen makes your pain and fever just disappear. And finally, Part 5, the plasma-derived complement system. We've covered the cellular ground troops. Now it's time to call in the big systemic reserves. The complement system is basically a series of dormant proteins just floating around in your blood, waiting to cascade into a lethal weapon. It mainly activates via two pathways. You have the alternate pathway, which is triggered by the IgA antibody, and then you have the classical pathway, which is triggered by IgG and IgM antibodies. A super easy way to remember this is the mnemonic General Motors. General Motors makes classic cars, right? So IgG and IgM trigger the classical pathway. Now, regardless of how it starts, it kicks off a very deliberate enzyme cascade. First, the trigger activates an enzyme called C3 convertase. Then, C3 convertase builds into C5 convertase. Finally, C5 convertase takes this dormant protein called C5 and essentially cleaves it right in half. This releases our ultimate biochemical hero, the active C5b molecule. And let me tell you, once C5b arrives on the scene, things get violent. C5b instantly fuses with complement C6, C7, C8, and C9. Together, they form the MAC, the membrane attack complex. This structure is quite literally a microscopic drilling machine. It slams into the side of a bacteria and violently drills thousands of physical holes into its cell membrane. Water then rushes into those holes until the bacteria swells up and literally pops like a water balloon. It's incredible. To ground all this in clinical reality, we have to look at what happens when parts of this system are naturally missing. For instance, a C2 deficiency is the most common complement deficiency overall. But if you have a genetic MAC deficiency, meaning your body can't form those microscopic drills we just talked about, you become highly susceptible to infections from capsulated organisms, specifically severe Neisseria infections. And if you're deficient in C1 inhibitor, which is the protein that's supposed to hit the brakes on this cascade, your body can't turn the swelling off. This leads to severe, life-threatening angioedema. And that brings us to the ultimate question. We've seen how histamine, cytokines, the arachidonic acid cascade, and the complement system all perfectly orchestrate our survival against external threats. It's this breathtaking, incredibly complex army keeping you alive every single second of the day. But what happens when the targeting system glitches? What happens when this heavily armed microscopic military turns its drills, its fevers, and its swelling entirely on us? Well, we'll explore that terrifying reality when we dive into autoimmune diseases in our next explainer.